in the Krebs cycle where energy is formed. Yeah, where energy is formed. It's either in the NADH or FADH2 or GTP, right? So indicate where in the cycle the energy is formed. All right. Okay. Also, number 4, you focus also the steps that involves production of carbon dioxide. Okay, remember the waste product of aerobic respiration is carbon dioxide. Okay, one is formed in the link reaction where the carbon dioxide is formed in the Krebs cycle. Focus also on that part. Okay, so that are 4 things that is important for the Krebs cycle as we go along, right? Keep in mind on that. Again, and then at the end of the lesson, you should be able to calculate how many ATP total produced by aerobic respiration, including the Krebs cycle. Okay? All right. Now, we continue back to the Krebs cycle. Okay, there are 8 steps in the Krebs cycle. Okay, let's go. The first one. Okay, this is an overview of the 8 stage of the Krebs cycle. It looks quite. Is it okay? Can you see the steps? Okay, or we can just go through this one lah, Encik saya. This one look more clear on the stages. Okay, go through this one first. Okay. Step 1, can we see step 1? Step 1 is oxaloacetate, form citrate. Okay, and then citrate will be isomerized to form isocitrate. Okay, and then it will be converted to alpha ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and then get back to its oxaloacetate. Right, then the cycle restart. All right, okay, let's go through the details on this Krebs cycle. All right. Okay, if you can see from the diagram, okay, the Krebs cycle, keep in mind, it also occurs in the matrix of mitochondria. All right. Okay, now focus at the first point where acetyl-CoA from the link reaction, right? The acetyl-CoA from the link reaction will combine with the oxaloacetate. All right. Acetyl-CoA is a 2-carbon compound. Oxaloacetate is a 4-carbon compound. Right. Oxaloacetate is already there in the matrix, waiting for the acetyl-CoA to start the Krebs cycle. All right. Okay. So acetyl-CoA combined with oxaloacetate in the condensation process. Okay. Step 1 is condensation process. Acetyl-CoA, a 2-carbon compound, react with the oxaloacetate, a 4-carbon compound, with the help of an enzyme here named citrate synthase. Right? Citrate synthase to form a 6-carbon compound known as citrate. Right. The first step involves condensation of oxaloacetate to acetyl-CoA. Okay. Citrate, if you can see, it does not have the combined with the acetate, the oxaloacetate with acetyl-CoA will later cut the CoA out and left only the 6-carbon compound is known as citrate. All right. Okay, the first step. What is the process involved? Condensation. All right. Now, the 6-carbon sugar, the 6-carbon compound, the citrate, will be converted to isocitrate. Okay, citrate and isocitrate, they are isomers. right? They are isomers. Look at the arrow there. You can find a hydrogen removed and ester added. All right. So here, the reaction involved is dehydrogenation and rehydrogenation of citrate. Okay, look at number 2. Citrate is converted to its isomer, isocitrate, by removal of one water molecule and addition of another. Right? Okay, so here the reaction type is dehydrogenation and rehydrogenation. Okay, step 1 and step 2, tak ada lagi pembentukan CO2, betul? Tak ada lagi pembentukan ATP. Alright. Okay, so we are going to proceed to step 3, which is one of the most important step in the Krebs cycle. Alright. Look at number 3 in the cycle. Isocitrate is oxidized, right? Isocitrate is oxidized. Use the term oxidized here. Yeah, don't say isocitrate is converted. No, we cannot accept that. Right? So isocitrate is oxidized to alpha ketoglutarate. Right? Here, one NADH molecule is reduced to NADH. Yeah? NAD plus molecule is reduced to NADH plus H plus. Remember the H plus there. Okay. Kalau NADH, it will need only one H, but FADH2, it will take all the two hydrogens. All right. Okay. That is the first one. Okay. The reduction of NAD plus to NADH. Also, there is a decarboxylation occur there, isn't it? If you can see the arrow forming CO2 there, so that is the decarboxylation occur in the process, the formation of CO2. Now, isocitrate, 6 carbon compound. When it removes one of the carbon, it now becomes alpha ketoglutarate, which is a 5 carbon compound. Okay? Right. So now in step 3, one type of energy is formed, which is NADH. Okay? Right. So the reaction type for number 3 involve oxidative decarboxylation. Right? Number 3 involve oxidative decarboxylation. Means that there is an oxidation reduction process where NADH is formed, and then there is a decarboxylation process where CO2 is formed. Okay, class? For number 3, the reaction type is oxidative decarboxylation. Okay, now one type of energy in the form of NADH is formed in step 3. Okay, but now we are in the 4th intermediate, which is alpha ketoglutarate. Right. What happened to alpha ketoglutarate? It will undergo process decarboxylation again. Yeah, remove one molecule of carbon dioxide. Now alpha ketoglutarate is a 5 carbon compound. When it removes one CO2, it will become succinyl CoA. Yeah, succinyl CoA. This here now is a 4 carbon compound. All right, when it removes one CO2, now it becomes 4 carbon compound. Right, succinyl CoA, as the name is, if you can see the row there, alpha ketoglutarate after a decarboxylation will pick up one CoA. This coenzyme A is already there also in the matrix. Okay, it will combine with the molecule to form succinyl CoA. So, yang pertama, decarboxylation of alpha keto forming carbon dioxide. Right. And then the addition of COA to the compound forming succinyl-CoA. Right. Of course, here also there is an oxidation reduction process where NAD plus is reduced to NADH and H plus. Right? Okay. So if you can see now, step 4 is another step that form NADH in the cycle. Right? Okay, now we read number 4 there in the slide. Okay, number 4, another CO2 is lost through decarboxylation. And the resulting compound is oxidized, reducing NAD plus to NADH. Right? The remaining molecule is then attached to coenzyme A by an unstable bond forming succinyl-CoA. All right. Okay. Now, succinyl-CoA is a 4-carbon compound. Okay. Next, in the step 5, what happened to succinyl-CoA? Right? Succinyl-CoA will displace. Yeah, it will displace the CoA. Tadi dia pick up dekat step 4, and then now dia displace, buang balik, ya, buang balik the CoA, and then tinggal lagi lah 4 carbon tu je, 4 carbon compound tu, which is now called succinate. Yeah, okay. At the same time, one phosphate molecule also there in the matrix is added to GDP. Nampak tu? Okay, PI is added to GDP to form GTP, right? Okay, so here the process is okay, so one phosphate molecule attached to combined with the GDP to form GTP. All right, this GTP will later form ATP, where the phosphate from the GTP is added to ADP to form ATP. Okay, student, if you can see now, GTP is the substrate, right? When the phosphate from GTP is taken to combine with the ADP to form ATP, here the process is substrate level phosphorylation. Yeah, so GTP is considered now the substrate. Yeah, when the P is taken from the substrate to give it to ADP to form ATP, here the process is substrate level phosphorylation. Okay, now look at number 5 in the diagram. CoA is displaced by a phosphate group. Dah bincang tadi, alright, which is transferred to GDP forming GTP. Okay, GTP, you know already what it stands for, right? Okay, what is GTP? Is a molecule which functions similar to ATP. GTP can also be used as shown to generate ATP. Alright, okay, so that is number 5. Okay, cikgu rumuskan lagi sekali what is happening in step 5. Ada 3 perkara. Okay, the first one is the displacement of coenzyme A from succinyl-CoA. That is the first one. Second one, the addition of a phosphate group to GDP forming GTP. That is the second one. Okay, and the third one, the GTP will give the phosphate to ADP to form ATP. Here, the substrate level phosphorylation occur. All right. Okay, now step 5. Succinyl-CoA with a 4-carbon compound is changed to succinate. All right. Now we are in the next intermediate, the succinate. Okay. Now what happened to succinate? If you can see, the following steps of the Krebs cycle, the number of carbon will not change from succinyl. Succinyl ada 4. Next one is succinate, also 4. Fumarate, also 4. Malate, also 4. And back to oxaloacetate is also 4. Okay, it means that the next steps of the Krebs cycle does not involve the process of decarboxylation. Right? So decarboxylation only occur in step 3 and step 4. Right? Okay. Right. Now we move on to succinate. All right. Succinate will be oxidized. Nampak tak? Kenapa cikgu kata perkataan oxidized? Okay, dia keluarkan hydrogen and electrons dia. Okay, then hydrogen and electrons is being picked up by FAD to form FADH2 there. Alright, so here FAD is reduced. Okay, watch the term. FAD is reduced to FADH2. Succinate is oxidized to fumarate. Nampak tak perkataan oxidize and reduce is used differently here. Okay. Okay, now look at number 6. Kat situ written, the 2 hydrogens are transferred to FAD, forming FADH and oxidizing succinate. Right? So cikgu repeat here. In step 6, succinate is oxidized to form fumarate. And FAD is reduced to form FADH2. Okay, sekarang if you can see, there is another step for the production of the energy. Now, it is in the form of FAH2, right? So now if we can reverse back step 1, 2, 3, 4, 5, 6, you should be able to indicate where the energy is formed, right? So it is formed in step 3, step 4, step 5, and step 6. Right? Okay. Okay, succinate, a 4-carbon compound, oxidized to fumarate. Now we continue with the step 7. Step 7, fumarate. What happened to fumarate? It will be hydrated. Okay, nampak tak? The addition of water molecule. So addition of water molecule rearrange bonds in the substrate or the fumarate to form malate. Okay, so fumarate and malate, they are only they are not isomers, but the water rearrange the molecules. Yeah. All right. Okay. Now, malate, a 4-carbon compound, is oxidized. Is oxidized to oxaloacetate, and here NAD plus will be reduced to NADH plus H plus. Right? The process here is oxidation of malate. Right? Okay, where the substrate is oxidized, reducing NAD plus to NADH and regenerating oxaloacetate. All right. So the Krebs cycle is completed with 8 steps. All right. Students, are you okay? Okay, now, in the Krebs cycle, what you have to observe is, what you have to be able to explain is, the first one is all the steps involved. All right. So it consists of 8 intermediates. Right. So all the 8 intermediates be able to give. Right. Be able to give all the 8 intermediates. All right. And then how you get the name of intermediates if you are when you study and then you are familiar with the Krebs cycle, you can see the number of carbon only change from first step, okay, third step, and fourth step. Okay, and then later the number of carbon will remain 4. Right? Okay. Right. First, remember all the intermediates involved. Second, remember where indicate where decarboxylation occurs in step 3 and step 4. Okay, and then be able to indicate where in the cycle the energy is formed. So NADH is formed in step 3, step 4, and step 8. FADH2 is formed in step 6. GDP, forming GTP later converted to ATP, formed in step 5. Right, that's about it on the Krebs cycle. Okay, Encik Isa, you want to add anything?
Puan
Encik Isa,
Maziah,
you there?
Puan Maziah, yang
Yes.
ni slide terbaru ke yang lama punya? Ada yang terbaru yang dari Telegram tadi pagi.
Yeah, this is the new one from the
Cuba
Telegram.
pergi next page.
Okay.
Next page dia
Okay,
akan
this one,
explain one by one.
goes step by step, yeah. Okay, now let's just go through step by step the process. Okay, step one. Remember, here is the condensation process where oxaloacetate forming citrate. Oxalic acetate, 4 carbon compound. Forming citrate, a 6 carbon compound. Kenapa 6? Because acetyl-CoA 2. Oxalic acetate 4. So citrate is 6. Alright. Okay. Berlaku juga pada step 1 ini ialah the removal of coenzyme A. Yeah, it's set free and later should be used in the next turn of the link reaction. Okay. Okay, oxalic acetate forming citrate. And then in step 2, removal of water and addition of the water. Means here dehydrogenation and rehydration. Sorry, dehydration and rehydration of the citrate. Okay, let's see. Okay, itu nota dia. Nak tulis ke, nak snap ke? Condensation process, acetyl group of acetyl-CoA undergoes condensation with oxaloacetate and water to form citrate.
Class,
Citrate, yeah.
siapa? Okay, class. The module will only be given to you when the school reopens. Okay. So now, at the meantime, all right, I suggest that you self-study. Okay, so maybe you can snap or take picture of this screenshot. All right, do your own notes when you do your self-study. Okay, and then later whatever notes or anything that you have add in into your studies, you can add in into the module when you receive it when the school reopens.
Okay, tadi masa teacher describe this quite long, yeah. Teacher focus on this. I hope all the students take note on the name of the intermediates and everything that I have asked you to focus on. Okay, so in your note now, let's see the detail. Okay, add in some more details in your notes. All right. Okay, that is for condensation. The enzyme involved is citrate synthase. Okay, kat sini tadi. Add in now the citrate synthase to catalyze the formation of acetyl-CoA, combination of acetyl-CoA and oxaloacetate to form citrate. Alright, the next 7 step decompose the citrate back to oxaloacetate. Kan? Dia daripada oxalic acetate 4. Okay, citrate 6. So the next 7 steps decompose back citrate into oxaloacetate. Daripada 6 kita nak jadikan 4 carbon compound balik. Faham? Alright. Okay. Next step is dehydration and rehydration of citrate kan. Step 2. Right. Citrate is isomerized. Nampak tak ayat tu? Citrate is isomerized to isocitrate. How? By dehydration. Dehydration, as you can recall, That is the removal of one water molecule. And later, rehydration occurs, the synthesis of one water molecule of citrate. So citrate now form isocitrate, right? With that two process, dehydration and rehydration. Okay, step 3. Okay, kat sini apa tadi cikgu sebut? Oxidative decarboxylation. Why? There is an oxidation reduction process and also there is a decarboxylation process. Right? Okay, so isocitrate undergoes oxidation and then decarboxylation to form alpha ketoglutarate and releases CO2. Right? Okay. The H plus from the oxidation process of isocitrate is used to reduce NAD plus to form NADH. Okay, student, tengok ya. Kalau NAD plus, bila dia form NADH, we said is reduced. Alright, so it's reduced here in the meaning of it accept one hydrogen molecule. Okay. Alpha ketoglutarate. Be able to spell it correctly when you describe the Krebs cycle. Okay. The name of the enzyme. Also, you have to remember the name is isocitrate dehydrogenase. Right? That will catalyze the reaction of isocitrate converted into alpha ketoglutarate. Okay. So
Puan Maziah, siapa?
reaction process that produce FDH. Yeah.
Okay, class, always remember NAD plus and FAD, okay, these two compounds, they are hydrogen and electron acceptor. And every time when they accept either hydrogen or electron, okay, it has to be in a pair. So if they receive hydrogen, it has to be two atoms of hydrogen. Sorry, two ions of hydrogen. Meaning to say 2H plus, right? And a pair of electron, meaning to say two electrons. So every time this NAD, okay, NAD plus accept hydrogen ion, so it will form NADH with H plus. Okay, the H plus, the addition of H plus, it tells you that there are two hydrogen ions here. And as what I've mentioned earlier, the FAD, okay, when it receives the hydrogen, it becomes FADH2, right? And then with two extra electrons. And then the two extra electrons, either from the NAD plus when it receives the hydrogen ions, or the FAD when it receives the hydrogen ions, this pair of electron will be transferred to the electron transfer chain. Later when we reach that part, you have better understanding. Okay.
Okay, that also explains why NADH will give three ATP, but FADH2 only produces two. Okay, that the number of electrons involved explains that later in electron transport chain. Okay, thank you, Cik Isa. Okay, so we continue this in step 3, where isocitrate is oxidized, reducing NAD plus to NADH. That is the first thing to occur. Then the resulting compound loses a carbon dioxide molecule through decarboxylation process. Okay, now bila carbon dioxide produce, kita dah ambil satu carbon. So isocitrate, the 6 carbon compound will be changed to alpha ketoglutarate. Sekarang dah jadi 5 because it has been decarboxylated. Yeah, one of the carbon is decarboxylated. Dapat ya? Alright. Okay, next. Step 4. Step 4 kat situ sama. Betul tak? Ada NAD plus converted to NADH plus H plus. Ada CO2 form. Maknanya kat sini prosesnya ialah oxidative decarboxylation. Ya, oxidative oxidation reduction process. So decarboxylation, which is the removal of carbon dioxide. Okay, so 5 carbon compound alpha ketoglutarate. Right? Cikgu baca nombor 4 kat tepi tu. Another CO2 is lost. And the resulting compound is oxidized reducing NAD plus to NADH. Right? Then the remaining molecule, which is now 4 carbon, yeah, is then attached to coenzyme A by an unstable bond. Okay, so perkara yang pertama decarboxylation, yang kedua reduction of NAD plus to NADH, and yang ketiga the attachment of coenzyme A to the alpha keto the 4 carbon compound to form succinate CoA. Right, so be able to describe everything, all the reaction that involves here. Yeah, so there are some question asking you on Krebs cycle, they focus only where the energy is formed. Right, so here you should be able to describe in great detail what is happening in step 3 and step 4 and all the steps that involve the production of ATP. Okay, baik. Number 5, ni kat atas dia semua dah nota dah bagi tu, where alpha keto undergoes decarboxylation, succinyl attached to coenzyme A, then alpha ketoglutarate dehydrogenase is the enzyme involved, and then now is in the form of succinyl CoA. Alright, okay. Step 5, cikgu baca tu, CoA is displaced. Tadi dia masuk kemudian buang balik. Ya? CO is displaced by a phosphate group which is transferred to GDP. Okay. Bila GDP dia terima satu phosphate, dia akan form GTP. Right? This GTP is a molecule with function similar to ATP. Right. Okay, ATP and GTP memberi berfungsi benda yang sama, tapi it will be converted to ATP because ATP is more stable. Yeah. So GTP can also be used as shown to generate ATP. Okay, sini cikgu elaborate tadi berkenaan dengan substrate level phosphorylation berlaku kat sini. Okay. Kalau kita nak form NADH, the process is oxidation reduction process. Bila kita nak form ATP dekat step 5, it involves the addition of phosphate group to ADP. Yeah, so it's a process of phosphorylation. Okay, phosphorylation of ADP to form ATP. Okay, now phosphate group is from the GTP. Okay, so because of the GTP, now the substrate, now the process of adding phosphate group to ADP to form ATP is known as substrate level phosphorylation. Ini kat sini selalu pelajar miss, ya, dia tak ingat kat sini proses apa. Right, so substrate level phosphorylation. Okay, step 5 done. Okay, this is again there the substrate level phosphorylation. Yeah, succinate CoA displaced by a pi to form succinate. Pi transferred to GDP to form GTP. Then GTP donates pi to ADP to form GTP and ATP. Payah nak ingat, ada banyak tu, ada empat perkara berlaku kat situ. So setiap satu memberi satu markah lah. Yeah, succinate CoA synthetase. That is the name of the enzyme. Succinyl CoA synthetase catalyzes the reaction. Okay. Students selalu tanya, cikgu kena hafal ke nama-nama enzyme ni? Yeah, the answer is yes. If you are to describe the steps, you should give the name of the enzyme. Sometimes the question will not ask all about the Krebs cycle. It may focus to some of the steps. Yeah, if it asks on the step 5, then you have to mention lah the succinyl CoA synthetase is the enzyme that catalyze the reaction. Mungkin boleh beri satu markah kat situ. Kalau tak namakan, hilanglah markah kat situ. Okay, next step number 6. Oxidation of succinate. Tu. Succinate. Berapa carbon compound? 4. Semua bila dah sampai kepada succinate CoA, the number of carbon get back to 4 as what in oxaloacetate earlier. Yeah? So succinate CoA, succinate. Next is fumarate. Semua tu adalah 4 carbon compound. Maknanya bila dia maintain the number of carbon compound, it will not involve the process of decarboxylation anymore in all the steps. Yeah. Okay. So succinate kena guna ayat apa? Succinate is oxidized to form fumarate. Is oxidized to form fumarate. Gives out 2 hydrogen ion, as what Jesus said, and a pair of electron. Okay, so following the oxidation process is the giving out of 2 hydrogen ion, pair of electron, a pair. Right. Okay. Here, this 2 hydrogen ion is accepted by the FAD, later reduced to form FADH. So arrow kat situ FAD tu jadi FAH tu ayatnya ialah plus what is ya. Kalau NAD, kalau dia form NADH, dia ada NADH plus H plus. Nampak tak? Sebab dia ambil satu je hidrogen. Okay, tapi kalau FADH dia ambil kedua-dua hidrogen tu, so now luar. Faham kenapa? You senang lah ingat kat situ FADH2. Kalau NADH sebab ambil satu H ya, mesti kena tulisan tu tidak betul, tak balance lah. Okay, oxidase tu FADH2 lagi sekali formation of energy carrier. Okay. So hydration fumarate change to tak boleh macam fumarate is hydrated to but this is the name of the enzyme lah, the merase. To find awak tengok susunan okay, nampak tak macam mana double bond is broken. Water that kat melee, nak tak mana water molecule masuk. Alright, okay, so the water reaches the bonds in the fumarate by breaking the double bond, forming single bond in melee. Right? Okay. Okay, the last one is what happened to melee? It will be oxidized back to oxaloacetate. Yeah. Nama enzyme melee dehydrogenase. Oxidation will reduce NAD plus to NADH. Lagi sekali formation of NADH kat sini. Right? And then oxaloacetate is then recycled in the Krebs cycle. Okay? Ini kat bawah ni start. Oxaloacetate displaces the coenzyme A and attach to acetyl group from acetyl-CoA to form citrate, and the cycle continues. Sampai sini maknanya dia bila dia form the oxaloacetate, nanti akan datang lagi acetyl-CoA from the next another pyruvate molecule. Dia buat link reaction, dia akan buat cycle baru pula. Yeah? Okay. So that's almost all about Krebs cycle. Okay, next one. Kita dah nampak tadi, cikgu dah ulang 2 kali. Maknanya cycle tu cikgu dah ulang 2 kali ya. Okay, now kali ketiga. Kali ketiga kita fokus kepada number of carbon. Ya, cikgu juga masa cikgu explain the first one tadi lama kat slide tu, cikgu dah masukkan semua perkara dalam tu. Sekarang cikgu dah pecahkan satu-satu. Next one is for you to check the number of carbon. Okay, step 1. Oxaloacetic acid ataupun oxaloacetate lah. Oxaloacetate. Ya. 4 carbon. Alright. Plus acetyl-CoA 2 carbon. Citrate 6 carbon. Nampak? Okay. Dia sini dia isomerize. So 6 here, still 6 here. Okay. Sini decarboxylation step 3. 1 carbon. Dan alpha ketoglutarate ada 5 carbon. Okay. Step 4, nak decarboxylation lagi sekali. So, succinate CoA. From succinate CoA, succinate, fumarate, malate, and get back to acetate, tak ada removal carbon dioxide. Maknanya dah tak ada decarboxylation process. Number of carbon remain 4. Okey kelas, dapat tu ya. Okey, so kat sini nak letak NAD+, kita pendekkan. Lagi. Okey, satu formation of NADH, decarboxylation forming CO2, addition of CoA. Sini CoA keluar balik, succinate CoA tukar jadi succinate. Ya, sini ATP dihasilkan through GTP tadilah. Okey, sini FADH2 dihasilkan. Sini NADH dihasilkan. Okay. Kali ketiga dah kita nampak kat mana NADH dihasilkan. Satu, step 3. Yang kedua, step 4. Yang ketiga, kat step 8. Right? Hafal tu. Okay. Kemudian FADH ada dekat step 6. Yang ni tidak ada clue yang cikgu boleh bantu. Cuma awak kena hafal je lah. Ya, di sini antara succinate dengan fumarate ni ada pembentukan FADH2. Ya, okey. Kat sini GTP. So nampak dah di mana, mesti kena hafal. Kena tahu di manakah energy dihasilkan. Okay? Okay, let's see the next slide. Okay, so ini dah gabungkan semua proses kita. Krebs cycle tu dah habis, cikgu dah revise 3 kali. Okay, kita start balik the whole process of aerobic respiration. Start dengan glucose tadi. Glucose 6 carbon compound, isn't it? Okay, undergone glycolysis, forming 2 pyruvate. Right? This process, glycolysis, occurs in the cytosol. Pyruvate now enter the matrix of mitochondria. So this process sampai bawah ni berlaku dalam matrix of mitochondria. Alright, so 2 pyruvate change to acetyl coenzyme A. Ada 3 proses tadi ingat. Decarboxylation 1 and reduction process. Okay, and satu lagi oxidation dehydrogen of pyruvate into acetyl CoA lah. Ya, pyruvate 3 carbon form acetyl CoA 2 carbon. Right, this acetyl CoA enters the Krebs cycle where it combines with oxaloacetate and the cycle goes. Right, okay, nampak di mana penghasilan energi tu. Right, so ini nampak macam berserabut, tapi bila awak dah pergi 2-3 kali, awak dah nampak, awak dah rasa dah a bit more familiar with the name, isn't it? Okay, so tak ada banyak pun. Acetyl CoA 1 citrate, isocitrate, isomer dia. Okay, ini nama baru, alpha ketoglutarate. Kemudian succinate, fumarate, malate, oxaloacetate. So hafal 8 intermediate tu, hafal berapa bilangan carbon it has, then through the bilangan carbon it has, it will tell you where the decarboxylation will occur. Okay, okay, next slide. Now, this is the overview of cellular respiration. Cik sudah carikan ni. Okay, kita start balik dekat sini. Glucose, the process is glycolysis di dalam cytoplasm, forming ATP dan juga NADH. Okay, ATP ni dah siap dah buat ATP. Ada berapa ATP kita buat? Ada 8. Okay, 8. Ada juga dalam form ATP, ada in the form of NADH, ya, masa glycolysis. So what happened this to NADH? Dia mesti kena convert to ATP baru dia selesai kerja dia. Okay, nampak tak arrow ni? NADH zoom lalu terus ke proses yang dinamakan number 4 is electron transport chain and chemiosmosis. So next lesson, we are going to study the electron transport chain, that we are going to discuss what happened to this NADH in order for it to convert to ATP. Okay, so one NADH dia akan convert jadi 3 ATP. Okay, what is happening in the electron transport chain in order to harvest the energy from NADH to form ATP. Okay, baik. Okay, pyruvate daripada cytoplasm move into matrix of mitochondria. continues with the link reaction, where it will be converted into acetyl-CoA. Right, and then this acetyl-CoA enter the Krebs cycle, forming all these types of energy lah. Sama lah kat sini pun buat NADH and FADH2 tadi. Ni GDP dia terus convert jadi ATP tadi kan dalam cycle. Okay, NADH and FADH2 pun juga akan melalui electron transport chain. Dekat link reaction pun tadi kita bincang, dia buat NADH juga pun juga akan pergi ke proses electron transport chain untuk dapatkan energy from it. Ya, okay. Lagi sekali, kita tengok. Alright, pyruvate from glycolysis. Okay, ini link reaction lah ni. From glycolysis, pyruvate forming acetyl-CoA. Masuk citric acid cycle, dia bergabung dengan siapa ni? Oxaloacetate. Forming citrate, isocitrate, alpha keto. Nampak NADH dihasilkan kat sini, NADH dihasilkan kat sini, succinyl-CoA, succinate, fumarate, malate. So this diagram is to show you where in the cycle the energy is formed. Right here, and the second one, GTP, FADH2, and then from malate to oxaloacetate. Alright, okay. So here also is to tell you how many NADH is produced per cycle of Krebs cycle. So NADH buat kat mana? 1, 2, 3. Right. Okay, so 3 NADH. FADH2 satu je kat sini. GTP 1. So altogether ada berapa? 3 NADH, 1 FADH2 and 1 GTP. But students, remember that pyruvate from the glycolysis kita buat 2. Right? There are 2 molecules of pyruvate formed from 1 molecule of glucose. So another pyruvate will go through the cycle. One will go through the complete cycle producing this much of energy also. Right? So all these numbers we just times 2. So kalau question tanya how many energy produced in the Krebs cycle per glucose molecule. Okay, per glucose molecule awak kena cakap 3 kali 2 sama dengan 6 NADH, 2 FADH2, 2 GTP. But if the question asks you how many energy is produced, how many ATP is produced per cycle of the Krebs cycle, so you go by this number, 3 NADH, 1 FADH2, 1 GTP. Dapat ke tak tu? Okay, so kalau 1 pyruvate produce this much, kalau 2 pyruvate kali 2. Okay, baik. So sekarang awak dah boleh kira. Kita dah habis proses aerobic respiration. By now, you should be able to calculate the 38 ATP from the aerobic respiration. Okay, so show in your calculation ATP is formed in glycolysis, either in the NADH or ATP. Okay, how many energy is formed in the link reaction? How many ATP is formed in the Krebs cycle? Just show us how the 38 ATP is produced from 1 glucose molecule. Alright? Okay. So this one is your homework. Everyone do the homework. Tomorrow, we want to see we will discuss of how you are going to turn your homework. Is either through the Google Classroom, which is kind of difficult to set up at this moment. So usually we just ask you to splash your answer through the WhatsApp group lah. Okay, so we will discuss on this, but please do, student, show us how the 38 ATP is formed from the aerobic respiration. All right, now the next slide is on the question. All right, heart, kidney, and liver cells they are the organs that produces 38 ATP. Okay, so all the all anaerobic respiration in heart, kidney, liver cell will produce 38 ATP. But aerobic respiration in muscle cell produces only 36 ATP. Okay, so this one also we want you to Google up and as your homework, and then also send us the answer. Okay, explain why. In the muscle cell, selalu organ-organ lain bila berlakunya respiration, cell respiration there, it will one glucose molecule will produce 38 ATP. But in muscle cell, one glucose molecule can only produce 36 ATP. Where are the two? Okay, you explain why. All right. Okay. Number 3, the body cells. Body cells macam mana pula? Kalau heart, kidney, liver 38, muscle 36. What about our body cells? Okay, now this one, Cik Isa also want you to Google. Yeah. Okay. Part 2, that is the address of how you can find the answer lah for those questions. Dah snap ya, dah snap gambar. Okay. Okay. Kita sekarang dah pukul berapa? Kita sekarang pukul 11:49. Okay, so kelas kita over at 12:30. Okay, so let us go through the question first, and once we finish the question, we will off the Google Meet, and then we will let you do the homework question. Okay, number 1, test the standing. Okay, look at the cycle. That is the Krebs cycle. Atau nama lain dia apa? Citric acid cycle. Dua tu je nama yang selalu digunakan. Ya. Okay, starting with one molecule of isocitrate and ending with fumarate. What is the maximum number of ATP molecules that could be made through substrate level phosphorylation? Okay, so ini pun a student. Encik Isa, are you there?
Yes.
Cik Isa, this one we just ask student to snap the question or how? Is this the homework? Or we want to discuss it now?
Kita belah suruh mereka buat lah. Think kita jaman masa
okay.
ni. Mereka snap. Tapi alamak ada jawapan dah. Dah keluar jawapan. Aduh.
Okay, tak apa. Okay, so sekarang ni nombor satu tadi you all dah tangkap gambar belum? Tangkap gambar question, nanti Cikgu Isa akan bagi modul. Cuma untuk apa ni, modul tu tak tahu kita boleh jumpa kan in 2 weeks time. Okay, so if it is possible for you to snap the picture of the question, just snap it first and then convert lah macam mana cara pun ya ke dalam your punya folder, and then kita discuss jawapan dia later. Alright, okay, number 1. Okay, number 2, again on the Krebs cycle, look at this. Carbon skeleton for amino acid biosynthesis are supplied by intermediates of the citric acid cycle. Apa dia nak kata tu? Citric acid cycle ataupun Krebs cycle kita ni kan ada intermediates dia. Okay, the intermediates tu lah tu alpha keto, succinate, fumarate, malate, they are called intermediates. So statement kata carbon skeleton for amino acid biosynthesis ni kebanyakannya menggunakan intermediates ini. Because that is the statement. So which intermediate would supply the carbon skeleton for synthesis of a 5-carbon amino acid? Okay, so soalan ni tanya pasal bilangan carbon number. So itu sebab cikgu cakap tadi, dalam masa you study, you can tahu which intermediates, all the intermediates with the number of carbon it consists of. Okay, so kalau dia nak synthesis of 5 carbon amino acid, intermediates mana? Ini citrate 6, isocitrate 6, alpha keto 5, kan? Succinate-CoA 4. Okay, so adakah succinate, malate, citrate, alpha keto glutarate, isocitrate? Okay, yang ini dah tak boleh sebab mereka dah okay. Cari jawapan dia. Cari jawapan dia. Alah, dah sebelah jawapan tu dah, tak apa. Okay, next one. Same cycle. How many molecules of carbon dioxide would be produced by 5 turns of the Krebs cycle. 5 pusingan. Okay, itu sebab cikgu cakap tadi, masa you study, you kena tahu satu pusingan ada berapa CO2. Ada berapa tempat tadi? 2 tempat kan? Dekat isocitrate to alpha keto, then from alpha ketoglycerate to succinate CoA. So 2 molekul per cycle. Kalau kali 5, cek jawapan dia. Okay, next one. Next one, still on the same cycle. How many reduced dinucleotides will be produced with 4 turns of citric acid cycle? Okay. Maknanya dinucleotides ni dia refer kepada FADH2 and NADH lah. Okay, so dalam 4 complete turns of citric acid cycle, ada berapa FADH2, ada berapa NADH. Tadi kita dah kira, you all dah tahu nampak satu turn berapa, so kira je lah, kalilah dengan 4 turns. Okay, next one. Starting with citrate. Citrate mana? Okay, citrate. Starting with citrate, which of the following combination of products would result from the 3 turns of the citric acid cycle? Which of the following combination of products would result from 3 turns of the citric acid cycle? Okay, so maknanya dalam ni, dalam Krebs cycle dia tanya je lah, awak kena kira lah product would result from 3 turns. Okay, 3 turns, apa-apa yang kita buat dalam 1 turn sekali dengan 3. So that is the amount of ATP form. So kalau 3 turns tu ada berapa carbon dioxide form? Kalau 1 turn kan 2 carbon dioxide. Kalau 3 turns 6 lah. So jawapan dia antara dengan E. Okay, you find out the answer. Okay, next one. Carbon dioxide is released during which of the following stages of cellular respiration? Kan dah cakap tadi. Okay, so dalam Krebs cycle tu kena tahu adakah decarboxylation occur. Okay, so dia boleh bagi dalam bentuk Krebs cycle itself, dia boleh bagi dalam bentuk ayat macam ni. Kalau dalam bentuk ayat macam ni, awak nampak macam payah sikit, you have to draw or sketch the Krebs cycle lah, so that you can see where that one you can memorize straight. Glycolysis ada ke? Tak ada. Oxidation of pyruvate to acetyl-CoA and the citric acid cycle. Oxidation of pyruvate to acetyl-CoA. Ada. Macam ada je. And the citric acid cycle. Citric acid cycle and oxidative phosphorylation. Dia tak ke sini involve. Oxidative phosphorylation and fermentation. Belum belajar lagi. Okay. So you find that. Okay. So this is your homework. Everyone snap the question. Alright, for each molecule of glucose that is metabolized by glycolysis and the citric acid cycle, what is the total number of NADH plus FADH2 molecule produced? Okay, so yang ini dapat tak dia fokus only kepada NADH and FADH2. Okay, dia tak tanya ATP. Jawapan dia tak boleh kata 38. Kenapa? Sebab dia fokus kepada NADH and FADH2 saja. Alright, so carilah dalam glycolysis ada NADH, dalam link reaction ada, dalam Krebs cycle pun ada. FADH2 only appear in the Krebs cycle. So you total up everything, get the number of ATP produced from NADH and FADH2 per glucose molecule. Okay, the answer is E. Then you try to calculate. Siapa yang tak dapat jawapan E, kena fahamkan dulu. Kena fahamkan dalam cycle tu cari kat mana-mana. Okay, so for those of you don't get the answer E, please ask us. Dah habis. Okay, so that is the end of the today's lesson for the Krebs cycle. Okay, class, is everyone able to snap the question for your homework?
Class, turn on your mic so we have some response here. Okay.
Boleh snap tak? Boleh snap gambar? Cukup apa tadi? Sempat tak soalan? Soalan as homework. should I? Okay, yang ini soalan dia. Kemudian satu lagi, cikgu nak splash yang ni lah. The first one, yang ni. Any code. This one ada semua benda. Ya. You tengok gambar yang ni. I think this one is the most complete diagram of what is
Well, Maziah,
happening
you
in
know
the
what?
cycle.
You know what? Since somebody suggested to send to the group, you know what? Okay. Later I will screenshot every question and with the overview of the Krebs cycle. Okay, I will post it in our group, our
Mm,
WhatsApp group. All right. So
mm,
from there, I
mm.
think you should be able to do your self study.
Okay, so I think we should end here. Make sure tadi cikgu suruh buat 2 homework kan. Satu homework is on apa ya? Ingat dah. Satu homework, the calculation, the calculation of 38 ATP daripada mana dapat you buat dalam bentuk apa ni, jadual ke apa, tunjukkan macam mana 38 ATP is produced through all the stages, glycolysis, link reaction, and also the Krebs cycle. Okay, that is one homework. Second homework is on the ATP. Okay, why is muscle cell under 36 ATP? Why not 38? Where goes another 2? Right? Okay. So, Ansha Isa, you want to add in anything before we end the lesson?
Lapar mau pergi makan.
Just dimakan. Jom. Okay.
Okay, bye
Kita
everyone.
ada kelas Monday and Wednesday.
Wednesday.
Yeah? Follow
Wednesday.
6. All right. See you all Monday.
Bye everyone.
Bye,
Okay.
everyone.